we set up the experiment so that we were looking at treatment with either just regular media, growth media for the cell lines, mock treatment, which is sterile water, or the BX catalytic treatment. And we treated every 3 days. This is 90 days in to the experiment, so they've been treated on an ongoing every 3-day basis for 90 days. And what we're looking at here are the MRC9 cells, which are a normal lung counterpart. This is what they should look like. They grow adherent to the bottom of the well, and they're very long, very spindly looking. They're very happy in this photo. I don't see a lot of dead cells that are floating in the media. There's really no toxicity here to speak of. In this image, we're looking at mock treatment, which again is sterile water of the same normal cell line for lung. And again, you still see that spindly look. Don't see a lot of stuff floating in the media. There's really no toxicity. This is the BX treatment for the same normal lung cells, 90 days. I see the same sort of general morphology. I'm not seeing changes in the normal cells. Okay, they still look happy. They still are growing adherent. They still like the environment. The H2172 cells are a non-small cell lung carcinoma that we've also treated or not treated or treated with sterile water for 90 days. This is the untreated. So this is what they look like if I put them in the well and I keep feeding them media every 3 days and I don't passage them. So they are adherent cells. Again, they grow on the bottom. If you look here, this is them happily sticking to the bottom of the well. This dark mass stuff that you see is where they start piling up on top of one another because they've run out of flat surface, and so they just pile up and they grow three-dimensionally. And there's nothing to stop them because they have enough food and all of the environment is making them happy. This is the mock treatment of those same cells. Again, you have that three-dimensional growth in here, and you can see that there's lots of cells on the bottom. They're very happy in this environment. But this is treated with sterile water every 3 days. This is the BX treatment at 90 days, and you can see there are clearly, noticeably fewer cells in this well. There's a little bit of three-dimensional growth remaining. This was piled up just like the untreated until the BX began having its effect, and we started seeing a diminishment of those cells so that when I pull the media off and change it out, fewer of those cells remain. So this is affecting also not only that three-dimensional growth, but the adherence down here is still present. But if you notice, lots of these cells are balled up, which indicates that they're either getting ready to divide or they are releasing from the bottom and they're not very adherent any longer. I do not see a lot of ongoing cellular division at the same kind of rates that I see in the other wells. So we're starting to really, by 90 days, see a diminishment of growth capacity and the number of cells remaining when we treat with BX in this case. So that's really encouraging. We definitely need to look into the adhesion molecules. We need to look at, obviously, the mitochondrial, the energy production, and the cell growth potential within the cells, and that will be our next goal. So we've already started the treatment of those in flasks for in the same series, and we will sample those at multiple times, and we will be running PCR to determine what is happening within the cells. But this is just a really quick visual to show you it's definitely having a very strong impact, and not on the normal cells, just on the non-small cell carcinoma cells. And it's interesting, I don't see overwhelming toxicity. I don't see the cells just sloughing off in big numbers, but they slowly start growing slower and less, energetically and less prolifically, so that we have a reduction in the cell numbers without seeing a lot of cells coming off and floating around and being dead within the media, releasing lots of toxicity. Because as you kill cells, you produce toxicity within the system. So the lack of that means these cells are happier because they're not dead, they're not being toxified, and yet they're certainly not growing at the same rate that these cells are. I've worked with toxic chemicals and what you see is just this morass of dead floating cells, and there's nothing adherent anymore when you treat with a toxin. And I don't see that effect here. So it's clearly a very different mechanism. It's also very exciting to see gross morphological changes without that toxic side effect that would go through and just wipe out everything. Because if you can reconvert a cell back to a more normal phenotype, then you've eliminated the basis for the disease. It's going to be far easier for the body to tolerate. It's going to be far easier for the cells to take in and deal with. I just see so much potential for this because it is so different from any other toxic agent or anything else I've tried to treat cancer cells with. If you want to stop the cell growth, you can do that. You can treat them with a mitotic spindle inhibitor and boom. But then they're all stuck in one cell cycle situation, and before 24 hours is gone, they lift up off and they're gone. They can't survive. These are still living, but they are actually changing in their behavior, in their mechanisms of functionality, and that's really cool. I think it's fascinating. So it's not like anything else I've worked with.